The top of the hour. I want to welcome everybody to the special AIDS Day Symposium. We do this every year. We don't have it exactly on December 1st because our scheduled time is for Thursdays, and this year December 1st was a Tuesday. But we're really quite excited to have our three speakers today talk about COVID and COVID research, but really with a focus on how the past is prologue, how our experience over the last 35 years of studying HIV and other viral diseases has really prepared us for, on a scientific level, a very rapid response, an impressive response to the COVID epidemic. In fact, I would say it's 180 degrees different from the public response, which has been disappointing. But the scientific response has been amazing. So I'm not going to spend a lot of time on introductions for the topics, but I think it'll become clear as you hear each of our speakers how their HIV experience has transitioned to give us incredible insights into COVID and how we're going to be accomplishing bringing it under control from a scientific perspective. So our leadoff speaker is Dr. Paul Gebfert, who is a professor of medicine in the Division of Infectious Diseases and associate director for the CFAR. His experience ever since he was a fellow is in vaccinology, and he's going to talk to us about the COVID vaccine and where we are. And Paul, looking forward to your talk.
Okay, thank you. I'm going to share my screen. And is this working for everyone?
Yeah.
Okay, great.
Yep, looks good.
So I think the format for this is that the three of us are going to give us talks, and then we're not going to entertain questions until the end, if that's okay, unless there are any burning questions. And so I'm sorry. And so I'm going to talk about the development of COVID vaccines. And in particular, I'll just, throughout, I'll talk a little bit about how it is that the HIV field has actually helped in this rapid development of vaccines, which, as everyone probably knows, is sort of a historic achievement. I'll just say that the prior vaccine record for development of a vaccine is 5 years for the mumps vaccine. And while it's not, I guess we wouldn't still consider this fully developed, it's going to be pretty close, and it likely will be developed in about a year, which is phenomenal. So just a couple of disclosures, Sanofi and Altimmune, which I won't be talking about much. This is probably the most important slide for us and for me and for us, and that is to acknowledge members who have helped with this effort. So as you know, this started back in February, March, thereabouts, and all these people on this slide have contributed significant amounts of time to help with various aspects of it. And I know I probably have left some people off, but they have all spent a huge amount of time during COVID, and none of this would have been possible without each one of these individuals. Now, maybe the phase 3 studies in general, possibly for the COVID vaccine, since we're just now going to start those, but lots of other aspects these people have been involved with, and James and Nathan will talk a little bit more about that as well. So just before this all began, we have this large HIV vaccine trials network, and that actually, when COVID began, it morphed, it combined with the HIV prevention trials network. And this is just the HIV trials network, the number of sites that they have, and you can see it's a global entity. And they are able to, to have sites ready to go at all these places, and the HPTN, the same thing. And they've sort of merged into what's called the COVID the COVID virus prevention network, or the coronavirus prevention network. And so they were ready to go to start doing very rapid clinical trials. So just as a few things, everybody knows this, but it's just kind of remarkable to me. Every time I look at these numbers, you can see, I think it's 60 million now, but this was November 19th. And it's rapid, it's going very, very fast. And just yesterday, as many of you know, in the United States, it's more than 14 million cases, more than 100,000 hospitalizations, and about 3,100 deaths, which is, all of those are more than we've ever had. And I'm sure you've heard the stories, and the UAB is really being stressed right now by the number of individuals that need admission because of COVID. And so this, we're hoping to weather this storm. And then this is also some interesting data, just to put this into perspective. You can see that in the beginning it was dominated by people who are over 65 in terms of hospitalization. But then there, just under that, you can see younger age groups. And then a little bit younger age groups kind of increased, and most all of them are associated with some sort of comorbidity. And then you can now see more recently that the rates again are increasing. I just pulled this from the Alabama thing this morning. And these are the new cases, and I'm guessing that most of you looked at this, so I won't go too much into this. But you can see that, we're averaging over 3,000 cases in the last couple of days. And what's even more worrisome is the hospitalization rates are highest than they've ever been. And Thanksgiving, we haven't seen the impact of Thanksgiving yet, and so I think that's what has us worried. Okay, so now I do want to talk a little bit about the good news. And so I just want to give one sort of thing about vaccines. So we have a lot of vaccines, and a lot of them are shown on this slide. And what's important to know is that while the immune system does a bunch of different things to react against a pathogen, the main one that actually is the correlate of protection in many different vaccines that are already approved are antibodies. And neutralizing antibodies are what's extremely important in that situation. And so a neutralizing antibody is kind of defined as really a laboratory phenomenon, if you will, where you just put in the virus and the antibody, and if the antibody kills the virus, it neutralizes it. And those are sort of the best types of antibodies that you would like. And so very early on in this, people were wanting to get a vaccine for COVID-19, and they really wanted to target these neutralizing antibodies because historically these are sort of the best types of immune responses that you would want for a vaccine. So these are the vaccine candidates that are in development. And I know it's a busy slide, but you can imagine there's all sorts of different types that are being developed. I will say the ones that are either in phase 3 trials currently or are planning to start any day now for phase 3 trials are the ones that I show here in green. And I will focus on the Pfizer vaccine, the Moderna vaccine, and the AstraZeneca vaccine. And then our site just got IRB approved about an hour ago to start enrolling into the Janssen vaccine from Johnson & Johnson. So we'll talk a little bit about the messenger RNA vaccine, and messenger RNA is actually a novel technology. But what's nice about it is that it appears to be able to induce very good immune responses, and you can actually synthesize these things and manufacture them very quickly, which is why they were the first out of the gate, Pfizer and Moderna. The next easiest to make in sort of clinical grade is the non-replicating viral vectors, and both AstraZeneca and Janssen do that with the adenovirus non-replicating vector. Protein-based vaccine, which a good example of one that's clinically based right now is the hepatitis B vaccine. these are historically our best vaccines, tried and true technology, but the problem is it takes a while to optimize protein manufacturing in the test tube and then using that to inject into the participants. So these are taking a little longer, Novavax and Sanofi, and neither one have started clinical efficacy studies yet, although Novavax is supposed to do that any day now. So just to get to the good news, November 9th, this is a New York Times article. It shows that the Pfizer vaccine, back then they said 90% efficacy, but now it's 95% efficacy. All we know is what the companies are telling us. None of this is published right now, but this is just a phenomenal finding, in that we thought that at best we would be able to replicate what influenza does in the best years of influenza vaccines. So when the influenza vaccine matches what's circulating, 60% efficacy is what we get. And we were hoping for that, maybe 70%. Nobody really thought 95% was a possibility. And so you could say, well, maybe this was just a fluke. It was a 30,000, well, actually this was a 44,000 person enrolled study where half got vaccine, half got placebo. But then a week after that, Moderna, with a 30,000 enrolled study in the United States, showed essentially 95% efficacy as well. And you may, I'll just go forward with this, you may actually know that more recently, sorry, there's a siren going here. More recently, the Moderna company came out and showed that now they have almost 200 total cases of COVID. All but 11 are in the placebo group, and they had 30 cases of severe COVID, including one death, and they were all in the placebo group. So not only does it prevent symptomatic COVID infection, it appears to be preventing severe disease, which is fantastic news. And then finally, AstraZeneca came out, and AstraZeneca is the recombinant chimp adenovirus that can't replicate, and it expresses the same protein, the S protein. It's interesting, this was 90% effective in one area of the world, 62% effective in the other area of the world, and the larger numbers where the 13,000 people study was done was 62% effective. I think the take home at this point is that it's not as effective, it's 70% effective. And why that is interesting. We don't know yet. It is a different technology. That may be part of it. It's also something that a lot of people don't know is the S protein form in the Pfizer and the Moderna vaccine is what's called a fusion stable complex. So a pre-fusion stable complex that was developed by the NIH and Barney Graham. And let me just say that that's the Vaccine Research Center, which was actually was established during the time of the Clinton administration to deal with the HIV epidemic and pandemic and try and develop a vaccine for that. So again, this benefited from the HIV, the resources that we've put into HIV, because they knew immediately that this is what they needed to do, is this pre-fusion complex. That pre-fusion complex S protein induces higher neutralizing antibody responses, at least in preclinical studies. But for whatever reason, AstraZeneca decided to go with their own non-pre-fusion complex. And maybe that's doing something, but we don't know just yet. I wanted to talk a little bit about side effects. So almost everybody is going to have mild to moderate pain after the Pfizer vaccine. And you can see after one dose and after 2 doses, most everybody has pain. And I would like to say that this is true with the influenza vaccine too, so it's somewhat expected. And we'd like to say that this is meaning that the vaccine is likely going to work if you're feeling some sort of reaction to it. Systemic reactions also occur with these. And right now, Pfizer has gone, their efficacy study is in the 30 microgram dose. I should say that I didn't say that this is phase 1/2 studies. This was done in order for them to allow them to do the phase 3 efficacy study. But you can see in the 30 microgram dose, almost everybody has mild to moderate headache, which is interesting, and a great majority have fatigue following the Pfizer vaccine. And I should also say that Moderna is similar, maybe perhaps a little less. And I should also say that most all of these symptoms go away in about a day when they get them. So they're transient, which is good news. So this is really interesting. So this is the neutralizing antibody response that I was talking about. I highlight in red here the 30 microgram dose, which is being tested in the efficacy study. And what you can see is after 2 doses, these 2 arrows here, you get this level of antibody, neutralizing antibody response. So just this number 267. And what's amazing is it's higher than what you get in natural infection. So these are people who have convalesced from COVID infection. So the Pfizer vaccine gives you higher neutralizing antibody responses, as does the Moderna vaccine, than you do in natural infection. So that seems to be good news, and that may be suggesting why these vaccines work so well. So the AstraZeneca vaccine, I just wanted to show quickly that, again, it's similar types of side effects, not quite as many. Not everybody gets pain, but the majority do. Not everybody gets fatigue, but again, 60% do, and also headache. So it's a little bit better tolerated, but you still get some tolerability issues with this vaccine. The only other thing I want to point out is that the neutralizing antibodies that you get here are actually not, it's hard to compare between studies because not the same lab has done all this, but they take patients who've recovered and then they compare it to their individuals who've gotten a boost, and it's about the same. It's not higher, it's about the same as individuals who have recovered from COVID. So that's one possible reason why this vaccine maybe doesn't work as well. And I know this is all sort of conjecture, but these are things that people are looking at, scientists are looking at in the field. Just one slide then about the allocation plans of the COVID vaccine. The ACIP, a CDC organization that makes recommendations about who should get the vaccine. The phase 1A, they voted on this, so it's going to be healthcare personnel and residents of long-term care facilities. And this is sort of what's on the table for next is essential workers, followed by adults of high-risk medical conditions. But I think there's still going to be debate on that, whether that's the exact order going out. UAB is going to get their vaccines probably next week after the FDA convenes. We don't know exactly how many doses we're getting. The state of Alabama is getting 50,000 doses to go to 6 different medical centers. And we're obviously one of those. We have to go through the ADP, the Alabama Department of Public Health, for our vaccines. So just these are the conclusions, you can read them. I won't go through them. I will say that we are hoping now to start enrolling next week into the Janssen vaccine study. If anybody's interested in volunteering, let me know. However, this Janssen study has already enrolled almost 30,000 individuals. We only have 10,000 more to go. And that means we only need people who are not white, who are under 60. But if you're over 60, you can be any color. So if you fit that and you're interested, give me an email. Send me an email, please. And with that, I think I'll hand it over to Dr. Sag.
Yeah, thanks, Paul. Wonderful review and very timely, obviously. We're going to move on to Dr. James Kobe, who we're extraordinarily pleased that he's at UAB now. He's been here for a couple of years and came to us from Rochester. And he's been working a lot on monoclonal antibodies and the translation between the world of HIV and SARS-CoV-2. So, James, I'll turn it over to you to tell us what's happening with this. And this is in the realm of therapeutics.
Thanks, Mike, and thank you to the CFAR for the opportunity to present. Can everyone see my slides?
Yeah, you might want to put it into presentation mode, although we can see it fine the way it is.
Is it in presentation now?
No, that's okay. Just go forward. We're good.
We see your thumbnails on the side, but whatever you like.
Okay, well, in interest of time, we'll just go with this. So, I'm going to talk about the development of monoclonal antibodies for COVID-19 and highlight some ways in which efforts to understand the B cell and antibody response to HIV has really enabled the rapid and rational development of antibodies as therapeutics for SARS-CoV-2. My disclosures is that we are working with Aridis Pharmaceuticals. They've licensed some of the antibodies.
So the view we're getting is your sort of pre-mode, so you may want to reorient a little bit.
Okay, thanks, Mike.
Maybe go out and come back in again.
Yeah, unshare and go to the one that's presenting. Yeah, thanks. There you go. That's got it.
Okay, great. So yes, we're working with company Aridis Pharmaceuticals. They've licensed some of the antibodies we've developed and provided some funding to test the antibodies in animals. So as Dr. Geffert nicely discussed in relation to vaccines, antibodies are a major mechanism by which the immune system protects against pathogens. Antibodies are made by B cells, which are a type of white blood cell or a lymphocyte. In people that are infected with viruses, whether it's HIV or influenza or SARS-CoV-2, B cells that are specific against the virus respond, proliferate, and then begin to produce antibodies against the infecting virus. The antibodies that are most effective at inhibiting the virus or neutralizing it are those that bind proteins that are on the surface of the virus. Those proteins on the surface of the virus, particularly that are responsible for the virus mediating an attachment to the host cell. And by inhibiting these proteins, you can get inhibition of the viral life cycle and the viral infection. So over many decades now, by studying the B cell and antibody response to HIV in remarkable detail, the field has really revealed that the most effective target for HIV neutralizing antibody responses is the HIV envelope protein, also known as GP160. And the envelope protein is responsible for binding two proteins on the surface of the host cells, including CD4 and also the co-receptor. Now, there's different parts of the envelope protein that bind each. The CD4 binding site binds CD4, and the V3 region binds the co-receptor. The analogous process in SARS-CoV-2 involves the SARS-CoV-2 spike protein. And particularly the receptor binding domain of the spike protein binds to angiotensin converting enzyme 2 or ACE2, and also the S2 portion is responsible for making contact with a protease that further allows entry into the host cell. So really, developing antibodies that target these key proteins and the regions that come in contact with binding to the host cell receptors is really important to have a good neutralizing antibody response. So numerous antibodies have been cloned from people with HIV infection, and several of these monoclonal antibodies have been shown to be very broad and potent to recognize a wide range of HIV strains. And most of these before and currently still recognize the CD4 binding site as a primary epitope. VRC01 is one of these most fully studied monoclonal antibodies, and certainly the furthest along in terms of clinical development. And it shows that an antibody can be effective at suppressing virus. And so here is a study where people with HIV in this clinical trial, they stopped antiretroviral therapy and then they received VRC01 treatment or placebo. And it was shown that in HIV infected patients that the VRC01 treatments could reduce, although for a limited period of time, the amount of virus present in these people, and suggest that antibodies against HIV can suppress virus. This is a good start, but certainly needs more further development. Now, what's appreciated certainly from HIV and this leading to other fields is that a single antibody may not be sufficient to deal with all of the complexity in terms of HIV, the diversity in the virus strains. And so what's been determined is that a cocktail approach of antibodies that recognize different portions of the HIV envelope, such as the CD4 binding site, as well as the V3 region, that in combination, this cocktail may result in better ability to neutralize the virus and have broader recognition of potential differences in the virus. So with this kind of cocktail approach is what Regeneron has adopted for SARS-CoV-2. And they combined two antibodies that recognize different parts of the SARS-CoV-2 receptor binding domain portion of the spike. And then Eli Lilly has a single antibody drug. Now, what's been shown in these clinical trials is that both antibody approaches, the Regeneron and the Eli Lilly antibody approach, can reduce virus in the respiratory tract to some extent in the clinical trial participants that were treated with the antibodies. And then further, when these antibodies were tested in people that were had mild to moderate SARS-CoV-2, and particularly those people that were at higher risk of complications or leading to hospitalizations or emergency room visits, what was seen was in the Regeneron study that of the participants, 78 participants in the placebo group ended up having to have 7% of the placebo group ended up, 9% of the placebo group ended up having to be hospitalized, where only 3% of the Regeneron antibody cocktail treated group had to be hospitalized or have an ER visit. And there were similar results with Eli Lilly, where 15% of the placebo group ended up going on to hospitalization, whereas only 4% of the antibody treated group went on to hospitalization. As a result, these antibodies have been approved for emergency use authorization, but only in non-hospitalized patients that are at risk for advancing to severe COVID. What's really still unproven is the value of these antibodies in severe or hospitalized patients. This is certainly an important step in terms of a therapeutic for SARS-CoV-2, but certainly highlights the need for continued development of therapeutics for severe SARS-CoV-2 infection. So now for a few minutes, I'm going to touch on a little bit of what my lab is working on, as well as collaborators here at UAB and other institutions. So critical to developing and isolating therapeutic antibodies against SARS-CoV-2 is having good baits to be able to isolate those B cells that might be best at neutralizing SARS-CoV-2 virus. And particularly, we wanted to look at those B cells that are recognizing epitopes within the receptor binding domain that are specifically critical for ACE2 binding to the receptor binding domain. Mark Walter and UAB Microbiology developed recombinant RBD proteins, and then RBD proteins that are complex with ACE2, with the rationale that those epitopes that are responsible for RBD binding to ACE2 would not be available to antibodies to recognize or B cells in this complex formation, and then we could use these differentially to identify B cells that are specifically recognizing important RBD epitopes. Using these probes and the BSL2+ facility that was established by the UAB CFAR, as well as the generous development and receipt of a cell sorter, we're able to use these probes to isolate B cells against the SARS-CoV-2 receptor binding domain. Here is an example of doing that. Where you see on the right is a healthy control, where very few of the B cells of this healthy control recognize the receptor binding domain or the receptor binding domain that's complex with ACE2. However, in the COVID-19 patients, we see a greater frequency, although still small, of cells that can bind the receptor binding domain alone or also the receptor binding domain in complex with ACE2. We went ahead and isolated these receptor binding domain positive B cells, and using methodologies we developed for isolating HIV antibodies, made recombinant monoclonal antibodies from these B cells, and developed a panel of antibodies that broadly recognizes a variety of SARS-CoV-2 spike and receptor binding domain proteins, but have minimal off-target recognition of other antigens such as SARS-CoV-2 nucleocapsid. We went on to test these antibodies in collaboration with Luis Martinez Sobrito at Texas Biomedical Facility for the ability of the antibodies to bind to and neutralize SARS-CoV-2. We see that these antibodies, particularly this 1212C2 antibody, can bind SARS-CoV-2 RBD with high affinity and also neutralize SARS-CoV-2 in vitro at very low doses of 10 nanograms per mil. In red, you have, in comparison, the neutralizing activity and affinity of the Regeneron antibodies and an Eli Lilly antibody, and we see that this 1212C2 antibody appears to have some superiority in binding and neutralization to those. These antibodies were then tested for their ability to prevent and treat SARS-CoV-2 infection in the hamster model, where the antibody was given at 10 mg per kg either 6 hours before infection for prophylaxis or 6 hours after infection for treatment. What we see in red is this 1212C2 antibody, and we see, as compared to the controls, either of an isotype control antibody or PBS saline, we see that the 1212C2 antibodies have less virus in the upper respiratory tract as well as in the lungs compared to the controls. We also see that there is a reduction in lung pathology with the 1212C2 treatment. And then if you're looking therapeutic, and this is 4 days after infection, you see in the upper respiratory, in those 1212C2 animals, no detectable virus in any of the animals, but in the lung, only 1 of 4, in contrast to the controls where the virus is detectable. Also, so we saw a reduction in lung pathology with therapeutic treatment. So systemic delivery of an antibody, such as through IV or intramuscular injection, is not the most effective way to get antibody into the lung, where certainly an early infection is the predominant site, the respiratory tract, for SARS-CoV-2 virus. So we've worked with this company Aridis, who has experience with developing antibodies against infectious diseases, as well as developing antibodies for inhalation. And the thought is that by providing antibody through an inhaler or a nebulizer, more antibody would be able to get into the lung, but you also get systemic delivery in the GI tract and then recirculation, so systemic antibody resist. So we went ahead and tested this concept in hamsters again. In this case, it was just a therapeutic model where the hamsters were infected with SARS-CoV-2, and then 12 hours later were given the antibody in red, either as IP injection or in green through inhaled nebulizer in the cages. And what we can see is that we could use very low doses when inhaled, down to 0.6 mg per kg, looking at the lungs either day 2 or day 4, and see really still a suppression of virus. For example, if you look at day 4, all of the animals, the control animals, have virus in their lungs detectable, but none of the animals that are treated with 1212C2, including at this low dose of inhaled antibodies at 0.6 mg per kg, do we see detectable virus in the lungs. For reference, the Regeneron and Eli Lilly's antibodies are given somewhere between 30 milligrams per kilogram and 100 milligrams per kilogram when they're given through IV infusion. So with this promising result, this company Aridis is moving this antibody on to phase 1/2 trials to see if it's effective using self-administered inhaled treatment to treat mild to moderate COVID-19. And then my laboratory's efforts are moved towards developing an antibody cocktail. Now we've got this antibody 1212C2 that recognizes the receptor binding domain. We're working on developing antibodies that might complement this and recognize the S2 portion of the spike, which is actually quite conserved with other coronaviruses, including the endemic coronaviruses. And with that, I'd like to acknowledge the people who made this work possible, particularly in my lab, Michael Piepenbrink, Shangita Sarkar, Madhabanti Basu, Chris Bass, and Ruben Birch, and particularly the collaborators, including Mark Walters, Nathan and Paul, Olaf, helping to get the facility set up to work with these samples, and then at Texas Biomedical, Luis Martinez Sobrito and his laboratory. Thank you.
Thanks, James. Wonderful. Really exciting stuff, and remarkable in terms of the speed with which this has moved forward. Pretty cool to go from concept to the animal studies and now to phase 1/2. That's it's quite a bit of success. So congratulations on that. We're going to hold questions to the end. We're going to transition to Dr. Erdman, who has been kind of holding down the clinical trials fort in a big way at University Hospital, sort of keeping the large legal pad or whatever it is you carry around with you that has every patient. So we're anxious to hear an update from you on what you've been doing with your spring and summer vacation.
Yes, indeed. Can you hear me and see screen okay?
Yeah, we're good.
All right, fantastic. All right, so I'm going to talk about COVID clinical trials, specifically repurposing for this new threat, and try to at least touch on how we kind of leveraged what our experience was, what our infrastructure was for HIV, and some of the inpatient work we've been doing and trying to figure out how to deal with this COVID. Let's see if it let me advance. There we go. Couple minor disclosures, association with the CoVPN network. We have some funding from the School of Medicine, and they've given a couple talks for Bristol Myers Squibb, but that won't be relevant to what we're going over today. All right, so kind of the key to think about, which I think everyone has done to at least some degree, but everything is so much harder in this setting. So just trying to work through all the inevitable headaches that we deal with on every individual step of this process. So mitigating exposure for our team, mitigating exposure for our patients, limiting traffic on the wards, trying to figure out how to keep people healthy and yet do this science all in a developing landscape where what is safe and what isn't safe and how best to make all this work isn't necessarily clear. Everything that we do involving a patient involves PPE consumption. Everything therefore needs to be thought critically, is this really necessary or not? That makes it really tough on the clinical teams, but also on the study team, trying to make sure that we're doing everything that we need to do and nothing more. Every single participant interaction is challenged by the fact that there's a bunch of plastic and masks and gowns between us and the patient. All that's happening when patients are appropriately scared and intimidated, trying to figure out what's going on. Everything about collecting samples, processing samples, making all the logistics of the study work, everything is more tricky. So it's in this environment that we waded into dealing with COVID by trying to get trials and develop therapies. So I'm going to take everyone back in time. It seems like forever ago, and yet just a few months. It's one of those kind of paradoxes here. But I think the first time a lot of people in this audience actually heard about COVID, or what was the unidentified viral pneumonia, was back on January 9th when Dr. Friedman gave his lecture. There had been some early reports coming out in middle to late December that there was something going on. So this time in January, there was 59 cases. By the time we got to March 4th, that was the first time that I was digging back through my email that we had access to a protocol of a study that we were being asked to participate in. So this is a study that was a global effort trying to enroll high priority therapeutics for acute management. The very first patient of this study went on protocol in February at UNMC, and then sites were kind of rolling out from there. All right, so the platform here for this study is it's an adaptive, randomized, double-blind, placebo-controlled study, which is particularly relevant to think about as the Solidarity study came out just last night with the formal publication. So this is kind of the gold standard, but very intensive type of study, whereas there have been some other global efforts to try to get overcome some of the limitations by sheer numbers, and I think there's advantages to both. The primary outcome for this study was looking at recoveries at day 29. Every day that a patient's hospitalized, they require assessment, vitals, data capture, and there's follow-ups at days 15, 22nd, and 29. As mentioned, the initial kind of conversation started in very early January. The study opened by late February. So things that we had to start dealing with in a meaningful, tangible way was who the heck has COVID, which requires adequate testing, which fortunately we've certainly made strides on, although now with the current numbers that Dr. Gephardt reviewed, we're actually being strained on our ability to keep up with reagents. But just having a viable test that we knew how to interpret, we could get results in a timely fashion was not simply done early on. And it's a great credit to Six Leal and his team that rapidly scaled up. So then we had to figure out a way to screen patients. So that's great if someone has COVID. We had to identify them very quickly to be able to approach them for study and work with the care teams on making that happen. And we need to consent them, which sounds easy enough, but so do you do a paper consent? And if you do a paper consent, what do you do with the wet signature? Well, what about an electronic platform? But then you're bringing electronics in and out of the room. Well, how about a verbal consent? But then you need a witness. And where do you get a witness in the setting of when people aren't allowed on the wards? Everything gets so much more difficult, so much more quickly. This is going to be a study. There's going to be samples. So who processes them? Where do they get processed? How do you schedule that around the clinical trial? All the storage and documentation. And then we had to figure out a way to provide the product. This involves inpatient wards, in the ICU, again, dealing with PPE, entering and exiting the room. There's really no way to do any of these studies with one person. You need a team at all steps because once you go in, you're kind of stuck in there. We had to have medical supervision of this study, and this is in the setting of an outbreak where we didn't know exactly how everything worked. Lots and lots and lots and lots of data, and this remains a challenge. These people tend to get sick, and there's a lot of things that need to be recorded. And oh, by the way, there's outpatient follow-up. And really, at each one of these phases, we relied upon the infrastructure that had been built under Dr. Gephardt, Dr. Marazzo, and also the MSG group with Dr. Pappas on experience in conducting trials. And yet, those studies typically happen over months to years, whereas this was happening over days to weeks. So that timeline, what it looked like in practice. March 10th, we officially had a meeting to start trying to think about how we could do this study. Within a few days, the initial protocol documentation was sent over to the UAB IRB. And that's, again, that's not something that I was terribly involved with. That's a credit to Jennifer Wilson, Mary Bergeron, Terry Kennedy, and a lot of people making that happen very quickly. We had our, everything changed on campus on March 15th. We went to the limited business model, so all of a sudden a lot of our support staff weren't available, which again made things interesting. We had our virtual site initiation visit on the 24th, and by the 25th, we were officially open. At that point, the study was over a quarter enrolled, and we thought we might have 2 weeks to try to do this. So we had our team kind of thrown together. We're going to try to enroll in those 2 weeks, maybe up to 8, but really the primary target was about 4 enrollments. Again, I was digging through my email and I found this quote for myself. We have everything in place to enroll 2 participants tomorrow and we'll be infusing by midday, which was perfectly delusional in retrospect. It was so often the case with the inpatient work. We screened a whole bunch of people that first day, finally found one that was in window that had the testing at the right time. Of course, this patient ended up was on ECMO at the time. By the time we got everything squared away, we started giving drug at 6 p.m. And so we all were in for an adventure, and it only kind of escalated from there. So if you remember, when this is happening, we were at late March into early April. And so there was a rapid spike in cases that were happening in the community that was reflected in the inpatient numbers. And yet it's still very early on. So everything was a challenge. We had very, very limited therapeutic options, and when we did have someone that qualified study, we kind of felt a moral obligation to be able to have them engage on the study if possible, just because there weren't very many other alternatives to provide them something. Also, the way we were managing the disease at the time involved really aggressive intubation strategy, where if someone was escalating in their disease status and they were on 4 liters or more of oxygen, they quickly got intubated, which meant a lot of transfers back and forth. There were MET calls happening hourly on the wards, and we spent a lot of time in and out of the ICUs taking care of people that ended up getting very sick. All right, so by April 1st, we had our 6th patient enrolled, and our overall target had moved from 4 to 8. By April 3rd, we now had 8 people enrolled, but we are now targeting 12. By April 9th, we had our first outpatient visit, and we were bringing in a team from, again, the HIV side of the world. On April 19th, the study was already closed. We ended up enrolling 16 people at UAB. So that ended up being just over 3 weeks, and truly it was an all-day, everyday endeavor during that time. So on April 27th, the DSMB met for ACT and unblinded because of the difference between the placebo and the intervention. By May 1st, there was an EUA released for remdesivir. Based upon this EUA, that number is out of date. I think we hit 675 this morning, but there have been a tremendous number of moderate to severe cases of COVID that have come through, and we've treated that many based upon the work that was done in this initial phase. By October 22nd, last month, well, a month and a half ago anyways, remdesivir was the first officially FDA-approved treatment for COVID. And again, there had some really sick people, and our last patient from phase 1 actually left the hospital just a couple weeks ago after an epic hospital stay, and she's truly a pretty amazing person for hanging in there through all that. Again, this happened because of, and I wish I had more pictures, this is what I had handy to me, but there were a lot of people that put in unbelievable hours and truly committed to this effort to make this happen. There was no way we could compensate anyone based upon what their hours were. Like, this was all above and beyond because they cared and they were capable, and it's a credit to the people that had already had this experience with other trials to kind of be able to adapt and overcome this. Again, this is some of the data where the remdesivir showed benefit over placebo for patient recoveries. And this is kind of how this looks like in practice, of red being worse outcomes or more reliant on oxygen at the time of closure versus blue, which is recoveries, and some of the numbers that came out of this initial study. In parallel, all this is happening on the study side. We were again kind of building on our research infrastructure to try to learn more about the study. Very straight away, Dr. Geppert's team up in the CCB started enrolling convalescent patients. And then we started trying to build a system to figure out a way to enroll and capture samples from those in the hospital. It was a slow, very painful start of both Dr. Geppert and I running around the hospital trying to get people consented and then bringing blood back over. We had Drs. Kobe and Kutch and team members from all across fifth floor Bevel that had a paging system set up that we would come in and process as we got samples. We've now kind of built this out into a more routine process. We've enrolled just shy of 1,200 people, have over 500 sets of acute PBMCs and plasma, and supplemented this with samples from urine and oral saline rinses, and we're now kind of building an outpatient platform where we can kind of continue longitudinal work. And again, this is a huge credit to those that were on the clinical staff that allowed us to kind of make this work with them, because every one of these samples had to be drawn by someone who was already going into a room. We didn't have additional team to go in and collect because of inappropriate use of PPE. And then the folks that made this happen, running samples all over the institution. As Dr. Kobe and Gepper alluded to, some of this effort was immediately turned into data. And again, that's a credit to a lot of people that were making this happen on the fly. And some of this has helped describe what happens both in acute COVID, which not surprisingly turns out there's activation. But interestingly, when we look at longer term in the outpatient setting, there's increased changes in surface activation of different cellular compartments well after the recovery of initial symptoms. As was being alluded to for those that were on the call early, there's a thought that this may actually relate back to some of the phenotypes we're seeing in long COVID. The work continued on the ACT side of things. We immediately transitioned from Act 1 to Act 2, which was looking at some of the anti-inflammatory interventions targeting respiratory failure in COVID. This showed a benefit in the intervention of baricitinib, which is a JAK inhibitor, which shuts down some of the cytokine cascades associated with inflammation. This study was being conducted while the Solidarity study was released, and that's kind of changed things. And now the next phase is going to be comparing anti-inflammatory strategies. And we just finished completing enrollment of phase 3. So here's some of the data from the baricitinib Act 2, which shows, again, you have more blue, more recoveries with the intervention versus more red and fewer recoveries on the placebo side. And this is in the setting of already getting remdesivir. And this was particularly profound in those that had kind of more aggressive respiratory presentations in high flow requirement. The big question we now have is, well, this looks similar, although on a different scale than what was reported in not Solidarity, but in recovery. So how do we leverage our anti-inflammatory regimens along with our antivirals? And that's actually something we'll be looking at next. So I bring up ACT because that's the one that I've been involved with the most, but there are numerous other studies, and the intensity of this kind of adjustment has been the case for each of them. So ACT phase 3, by the way, the team that conducted phase 1 and phase 2, we enrolled 16 and 15 participants. We just completed phase 3 and enrolled 52 participants. The international study enrolled just under 1,000 total. Dr. Overton's team put together a really impressive inpatient effort and a focused amount of time to enroll for the canakinumab study. I was in name in charge of the 5001 study, which was a kind of a bio study of COVID disease, but truly that was run by Jenna and Shay, and they were phenomenal in taking care of executing that study. We had the hydroxychloroquine study, which took a ton of onboarding to try to build an outpatient platform for doing this work, involving drive-throughs and swabs and trying to make this happen. Dr. Heath has been working with anesthesia, trying to get this study enrolled, and then there's other studies that are happening now in the hospital or for outpatient patients with plasma, with Regeneron, and the Lilly monoclonal antibody. And while this has been happening, we've also been doing work on dispensing remdesivir in the most organized way we can manage. And right from the beginning, Drs. Heath and McCarty were coordinating our efforts on convalescent plasma. And again, every single one of these need to be built whole cloth in response to the developing pandemic. All right, so most important study or slide, and Dr. Gepper touched on many of these, but it's truly a credit to all the people here that this stuff has gotten done. We have continued to ask everyone to adjust and modify and roll with things. And the teams that have been established across this institution have responded. It's been really, really fun working with them. And there's all sorts of research happening in parallel as well. So thank you, everyone, for the chance to talk. And I look forward to any questions I might be of help with.
Bravo, all three. Wonderful discussion. I'll kick off questions, and then we can either put your questions in the chat, or you can just, as we normally might do, just kind of shout out. Paul, one of the questions that came up with regard to the mRNA vaccines, because we've never really had these before. The way it works, you make an injection, the mRNA gets transported into the cytosol of the cell, it starts producing proteins. Does anyone have a sense of how long the mRNA is stable and able to continue to produce spike protein once in vivo? Does it go on for weeks or months or just a few days? Do you know?
Yeah, that's a good question. Maybe James can answer that. I think it goes on for a while. It doesn't go away right away, but probably I would say a few days.
Yeah.
And there's some animal model data suggesting that. I don't think they've looked at that in humans necessarily. But mRNA is not the most stable compound that you can imagine, so it's not going to stick around forever.
Right, because I think the concern, at least that I had, was the opposite. What if it was stable and stayed there for weeks and months and continue to produce a spike protein? But I don't think that happens, right?
No. I mean, if you think about it as well, the immune response will get rid of it, right? Because what happens is the cells that actually are expressing this thing, the immune response will kill those cells. So,
and I guess the cells would have to have a -80 freezer to keep the fungal
right. Yes, exactly.
Okay. I have other questions, but I'll open it up to the group. Let's see, there's one that just popped in to the chat. Are there questions about how mRNA crosses placenta? What about vaccinations in pregnant folks? Have to ask your wife.
Well, Jody would be able to better answer that, the person who's asking the question. But I think it's a theoretical possibility. I think where studies in pregnant women need to go forward. I think that is a potential concern. And if it does, I mean, what would that mean? I don't know. Maybe then you'd vaccinate the child. I don't know, which is fine.
Yeah. There are a lot of groups that haven't been, I mean, what about people who've had it before and giving them a vaccine?
So most of these studies didn't outright exclude people who've had it before. The one we're doing right now is not excluding them. If you say, I had COVID, you still can be in the study. They've done serological surveys. We don't know the results of that yet, but there's a fair number of people in both Pfizer and Moderna who have evidence that they had COVID already. And so the big question, the big fear was making the disease worse, this antibody-dependent enhancement, or making it worse if you got the vaccine. And that hasn't happened to date. So I think it appears to be pretty safe.
Great. Mike Finnecore is asking, given the higher than expected efficacy with mRNA vaccines, what happens if somebody doesn't get that second dose?
You know, it's interesting. That's a good question. We don't know that. If you look at the neutralizing antibody, I wouldn't want to be the one to not come get the second dose, but I suspect it's going to give you some protection. And in fact, there was some rumor that the DSMB was saying something to that effect, but only time will tell to give us the true answer to that question.
Yeah. And then final question here from Sadeep. Have the vaccine trials included children? I don't think they have yet, but they're going to.
Pfizer has gone down to 12 years of age, so they are amassing that data, but they are certainly planning on even lower than that in a staged process in the near future.
Right. Letitia wants to know about an MIT study that had used AI that suggested that the mRNA vaccine may be less effective than those of Asian or African American descent.
I'm not aware of that study, so I can't comment on that.
But it seems like it's pretty universal. I mean, when you only have 11 out of, what'd you say, 11 out of 200, what was the number?
I mean, it's 95% effective. I can't imagine it's really that bad in Asians and Africans. And the other story is, why is it that the African continent is not really having ravages of the disease, which is a story for another time, but it's really interesting.
Right. Miriam wants to know, there's going to be a registry available that monitors the distribution after they roll out.
Oh yeah, they're talking all about that, and it's somewhat controversial, but yes, that will happen.
It's causing you pre-traumatic stress disorder.
Yeah, we had a big discussion on that, and some of the investigators weren't happy that they're going to be followed for up to 5 years, even without them knowing that they're being followed. So that.
Oh, maybe let's get a TikTok account and that'll do it. Hey, James, I'm just blown away by this notion of inhaled monoclonal antibody, and I'd not heard that before. Is that novel? Has that been used elsewhere?
There has been, I believe, 2 clinical trials previously for other indications, and I don't believe infectious diseases of inhaled antibodies. So it's not completely novel, and certainly you can think that, yes, it could be effective for SARS-CoV-2, but it could potentially be a method that's useful for any respiratory pathogens. So really thinking, the applicability for this and then beyond.
Right. Again, I'm just blown away with the speed and the efficiency with which your lab has pulled this together. It's really, I mean, for those who haven't been in the lab, that's quite an accomplishment. So congratulations on that.
Yeah, but it's not just my lab. It's just, we're in a great environment, with history of, already collaborating with people and resources that were readily available to us.
Right. Nathan, I've talked to you about this a little bit before, but I've been on a DSMB for the last 2 months on something called iSpy, which I'm pretty sure is different than your Act 1 and 2, but they're both adaptive trials. But iSpy is also working at UAB, and do you have a sense of where that is? These are just other kind of NABs and NIBs MABs that are being tested, and is that out of pulmonary, or where's that coming from?
Yeah, so that comes out of the trial platform actually came out of breast cancer, and that's what they use as kind of the protocol infrastructure, but it's being run out of the critical care setting. So it's an ICU specific study. Looking at my numbers, we have 44 that have been screened, and they've had 34 enrollments to date now. I think they've already rotated on and off a couple interventions, but they're looking more at kind of early phase 2 type reagents. So they started off with a panel of 5 high priorities, had a couple that didn't seem to have much signal, and been kind of setting up so that they can be in position to do later on phase 3 studies after they identify high priorities from these kind of rotating. So it's more similar to the recovery idea, where you have a bunch of people that are being compared to a baseline with a variety of interventions.
And we don't have time to do justice to it, but this whole notion of adaptive trial design is just fascinating in a very short nutshell. What's happening is that in real time, the data are brought in, and then they use these Bayesian probability assessments in real time. The computers give you these probability ranges, and if it looks like it's going in the wrong direction either way, it can declare early on whether they're continuing as futile or whether you've already graduated and it works and you don't have to fulfill the full thing. But we're so used to traditionally waiting for specific moments in time where you evaluate with a data safety monitoring board and then are approved to move on. This is happening in real time, and somehow it gets away from the Bonferroni adjustments. I guess they're modeled in there. It means that every time you look, you have to make it more stringent. Somehow they get through this statistically, and it's just been an amazing learning experience just seeing how this all goes on. Let's see. George Liu wants to know about what we know about SARS-2 in children in terms of incidence. Does anybody have their finger on the pulse of that? It happens for sure, and they don't seem to get as sick. My understanding is that people under the age of 4 are a little bit more resistant to initial infection, and they don't tend to get sick. There are exceptions to those kids, some of whom have died from even Kawasaki's type pictures and heart disease and that type of thing. And then there's a lot more skin manifestations, the so-called COVID toe and other things like that. But it's more, once a child gets above the age of 8, then they start behaving a little bit more like an adult. And certainly while they're adolescent, they're more or less just young adults and are really responsible for a lot of the transmission, especially picking it up in school and bringing it home. and the families get infected. We've only got a few minutes left.
So if I may, Michael,
Yeah.
absolutely. So so the problem is the studies did not focus on the infection in children. It's kind of omitted question in the whole COVID-19 research. But there's several good studies showing basically, as you mentioned, children above the age of 12 are infected with approximately the same rate as adults.
Yeah.
Teenagers. Children below 12 are infected a little bit more rarely. There's actually an extremely interesting article in Science which addresses something that we have been thinking about, suggesting that previous encounter with common cold coronaviruses, with the 4 circulating coronaviruses, can actually induce partial protection against the SARS-CoV-2. And we have no idea whether it is through antibodies, might be cell-mediated immunity. The fact that children, especially below the age of 12, as you know, they are just sick all the time, which is good for them, for building immunity. So they go through this, in schools, they go through this common colds pretty much every winter. That might actually provide partial protection in terms of symptoms. There's been an excellent study in Harvard, and it's not the only one, in which they compared the viral load in nasal mucosa in children hospitalized. So this is a little bit, we need to look at it in the context in which the study was done. They used to hospitalize children.
Yes.
Shockingly, the viral load was significantly much higher than the viral load of adult patient on ICU unit. So that suggests that children might be actually, more active in spreading the infection. And of course, most of them are asymptomatic for a longer period of time.
Right.
So in that context, reopening of schools is a very, very dangerous thing.
Right. And we don't have time to go into this, but a lot of the viral load persistence has to do with a disturbed initial innate immune response that also correlates to more severe disease in adults. Well, anyway, this has been a fantastic session. Thank you to our presenters. Paul had to drop off the call to join another Zoom call. I don't know if anybody's ever experienced that before, but it seems to happen. But thanks very much. This could be one of our best CFAR symposia. Very glad for everybody to participate. Everyone keep up the good work. Unfortunately, we got to batten down the hatches because this thing's going to be really exploding in the next 6 weeks. So everyone take care of yourselves and rest when you can. We'll sign off. Thanks.